Okay, in the last session we have discussed that is the nucleotides and the nucleosides composition. Okay, now in this session we are going to discuss about the structure of the DNA. So before going to the structure of DNA, we are going to discuss some of the historical background regarding about the DNA. So the DNA, it was discovered by one of the scientists. As DNA, it is a nucleic acid. The nucleic acid, it is formed by who is that person? The scientist, that is a Frederick Mister. It was discovered by Frederick Mister. Okay. The second, and due to the presence of acidic nature in the DNA, that is a nucleus, he named it as a nuclein or nucleic acids and to held. Okay. Then after that, these nucleic acids were named and termed by one of the scientists. Who is that scientist? By name Zacharias. The DNA, the term termed as Zacharias is the person who given the name as DNA. Okay, it was named by Zacharias, and the structure of the DNA, it is given by the study of with the help of X-ray diffraction technique. Okay, so it was studied by the two scientists, and they got the Nobel Prize for this work. Okay, so who is that person? That is a two person, Watson and Crick. Watson and Crick. Okay, so this is about some few historical background of the DNA. The DNA, it was found by the Frederick Mister, he who call it as, that is because of the acidic nature, as a nucleic acids, and the term given by as DNA, or who is that person, what we will call it as, that is a Zacharias. And also the DNA structure, it is given by the two person with the help of X-ray diffraction method. Who is that person, what we will call it as the Watson and the Crick. Hence the name of the DNA structure, it is the Watson-Crick model. Let us discuss the structure of the DNA. So the students, you already know that is the DNA, it is made up of what? That is the nucleic acids itself. It is made up of the three compositions. Which are those three compositions? That is a pentose sugar, nitrogenous bases, and also what we will call it as, that is a phosphoric acid, that is a phosphate group. Okay, so this DNA, it is a long chain of nucleotides. Okay, nucleotide, you already know the nucleotides, they are the composed of what? That is a pentose sugar, nitrogenous bases, and also that is a phosphate group. These grouped together to form a nucleotide. These nucleotides, they are join together. Okay, they join together to form a length. Okay, the lengthy chain, and these forms what? That is a DNA. Okay, that's why the DNA, or that is a or nucleic acids, we will define as the polymer of the long heteropolymer of nucleotides. Okay, is called what we will call it as, that is the DNA or nucleic acids. DNA, it is a long heteropolymer. Heteropolymer, why it is why we are using heteropolymer here? Because the hetero means different. The different chemical compositions are present in the nucleic acids. Which are those chemical composition? What is the pentose sugar, and also that is a nitrogenous bases, and also that is a phosphate group. So that's why it is also called as the heteropolymer. So these heteropolymers, they are joined together one by one by the formation of the bonds, that is the phosphodiester bonds. Okay, so they join together. So that's why they form a long chain. Okay, DNA, it is made up of a long polymer, heteropolymer of the nucleotides. If anybody asks what is the definition of the DNA or what is the definition of the nucleic acids, what is your answer? That is the nucleic acids are the long heteropolymer of nucleotides. Is it clear? Okay, next we are going to discuss about the actual structure. So how the structure of the DNA it could be, it is there. So you already seen in the blackboard. So I have written the diagram. Okay, so one by one we are going to discuss the structure of the DNA. Totally there are so many characteristic features of the double helical DNA, the double stranded DNA, it is there. So the few we are going to discuss in this chapter, in your syllabus it is included. Now I am going to give the discussion on that. The structure of the DNA, I already told, it was revealed. The structure of the DNA, the characteristic features of the DNA structure, it is given by the two scientists, Watson and the Crick. That's why hence the name, the Watson and Crick model of the DNA. In the examination they will ask, explain with the neat labeled diagram the Watson and Crick model of DNA. So whatever now I am going to explain, so you have to write and draw. Okay, let us discuss the Watson and Crick model of DNA. Okay, Watson and Crick model of DNA. So in the bracket you can mention, this is very important for the point of examination for the 5 marks. Okay, every year in the board exam they will ask. Okay, so explain the Watson-Crick model of the DNA. Draw a neat labeled diagram of Watson-Crick model of the DNA. Describe the neat labeled diagram of Watson-Crick model of the DNA. So they will ask. So this is no need to draw. If anybody interested to draw, this one also you can draw, but you have to draw. This is must and should. Okay, let me discuss. So here, before going to that is a Watson-Crick model of the DNA. The DNA, it is a double stranded or double helical structure to healthy. So it means that the first point, it is made up of what? That is a 2 strands. It is made up of what? 2 strands. It means that the DNA double helical structure, it is made up of, that is a 2 heteropolymer of nucleotides. So one it is this strand, one it is the back one. Okay, so the 2 strands, it is made up of what? That is a double strand. That's why it is called as the double stranded DNA. So the DNA, it is made up of what? 2 strands. That's why it is also called as the double stranded DNA. So it is made up of the 2 strands. Okay, and these 2 strands are anti-parallel. Okay, anti-parallel in the sense they run opposite. Okay, so later we will discuss. Okay, so here each 2 strands, they are made up of what? That is a nucleotide. The second point is that each strand is made up of nucleotides. Okay, so here this is the double helical model. So these two strands, this is one strand, this is another one strand. These two strands, they are made up of what? That is a nucleotides. Okay, so what are nucleotides? You are already familiar with that one. And the third thing is that what we will call it as, that is a these two strands are anti-parallel. Anti-parallel. So in order to understand what is anti-parallel, so let us look into that this structure. Okay, so this is the pentose sugar, and this is the nitrogenous basis, that is at the 2 carbon atom, and this is the third carbon atom. This is the 5th carbon atom. Okay, so this is one end. Okay, so if one nucleotide, it will joins with another nucleotide by the formation of the phosphodiester bond with the other sugar, where is that position, that is a 5th carbon atom. So 5th carbon atom ಗೆ attach ಆದಾಗ, okay, again the free end, it is a 3 prime, that is a 3rd carbon atom, it is the 3 prime end. Okay, so very next, so in the first that is in the other end, in the same chain, in the other end, the 5th carbon atom, it is the free end. That's why this is the one, that is a one direction it will run. The same in the opposite strand. Okay, so if that is a nitrogenous basis, it will bonds with the another nitrogenous basis of the opposite strand by the formation of the hydrogen bonds. So it will runs oppositely. Opposite in the sense, if we draw the diagram, that is a pentose sugar like this, that is oxygen molecule downwards, and the 3 carbon atom, it is here, and the nitrogenous basis bonds with another nitrogenous basis of the opposite strand. Here the 5th carbon atom, the phosphodiester bond, this phosphodiester bond bonds with another nucleotide of the 3rd carbon atom, and here the 5 prime end, it is the free end. Okay, so always that is the functional group of the pentose sugar. So ಯಾವುದೇ ಒಂದು pentose sugar ಅಲ್ಲಿ ಎರಡು functional groups ಇರುತ್ತೆ, that is this is the second and this is the third. These two functional groups, out of these two functional groups, the third functional group, it will bonds with the another nitrogenous basis. Okay, in this one strand, that is a 3rd carbon group, it is a free end. In this here, the 5th carbon group, it is a free end. In this opposite strand, this is 5th carbon group is free end, and oppositely, that is a 3 carbon group, it is free end. So if we draw the line, so this is the 5 prime 3 prime, and opposite again 5 prime 3 prime. So anti-parallel, in the opposite direction it will runs. That's why it is hence the name anti-parallel. The two strands are runs anti-parallelly. Okay, the next important is that we have to remember in this, that is the double-stranded helical structure, that is purines, they are always pairs with pyrimidines. Okay, purines pairs with pyrimidines. Purines pairs with pyrimidines in the sense, that is a nitrogenous basis of the one strand, it will pairs with the nitrogenous basis of the opposite strands. That is also based on, that is a purines always pairs with pyrimidines. It means that adenine, it is always pairs with what? That is a thymine, and guanine, it will pairs with always where? That is a cytosine. In case of it is, in case of the DNA, in case of the RNA, thymine, it is replaced by which one? Yes, that is a uracil. Okay, adenine pairs with thymine by the formation of the 3 hydrogen bond, that is a 3 bonds, 3 hydrogen bonds in the pair of there. Sorry, not it is a 3 hydrogen bond, it is the 2 hydrogen bonds. Guanine, it is always pairs with the cytosine. So with the formation of the 3 hydrogen bonds. Okay, adenine pairs with thymine by the formation of the 2 hydrogen bonds after one strand to the another strand. Guanine pairs with cytosine, so with the 3 hydrogen bonds. So purines always pairs with the pyrimidines. This is also called as the complementary base pair rule antanu sameta helತೀನಿ. Okay, very next, it is a fifth point is that is while you come to the diameter of the between the two parallel strands. Between these two strands, the length, the diameter, it will measures about, that is a 20 angstroms. The diameter, it is 20 angstroms. So between one strand and another DNA strand, okay, there will be a gap. That gap is what we will call it as the diameter. The diameter, it will measures about what? That is a 20 angstroms. Okay, and that is the one turn, it is about 34 angstroms. One turn is 34 angstroms. What it means, one turn? In this double helical structure, the DNA, it will twisted like a what? That is a what we will call as a twisted ladder. So if you stays andre ನೀವು ಎಲ್ಲಾದರೂ ನೋಡಿರಬಹುದು. Okay, so stays case ಗಳನ್ನ ಹತ್ತಬೇಕಾದ್ರೆ ಯಾವುದು ಒಂದು ಮನೆಯಲ್ಲಿ ಹತ್ತಬೇಕಾದ್ರೆ ಆ ತರ ಟ್ವಿಸ್ಟ್ ಆಗಿರುತ್ತೆ. So like that, in the same manner, the DNA, it will be twisted like, that is a twisted ladder. ಏಣಿ ತರ, ಏಣಿಯನ್ನ ಹೇಗೆ ನಾವು ಟ್ವಿಸ್ಟ್ ಮಾಡಿದಾಗ ಹೇಗೆ ಕಾಣಿಸುತ್ತೋ ಅದೇ ರೀತಿ DNA ಸ್ಟ್ರಕ್ಚರು ಕಾಣಿಸುತ್ತೆ ಎಕ್ಸ್ರೇ ಡಿಫ್ರಾಕ್ಷನ್ ಅಲ್ಲಿ ನಾವು ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ನೋಡಿದಾಗ ಅಂತ ಹೇಳ್ಬಿಟ್ಟು. ಓಕೆ, ಸೊ ಹಿಯರ್ ದಟ್ ಈಸ್ ಆನ್ ದಟ್ ಟ್ವಿಸ್ಟ್ ಮಾಡಿದಾಗ, ಸೊ ವಿ ವಿಲ್ ಸಿ ದಟ್ ವಾಟ್ ದಟ್ ಈಸ್ ಅ ಟೂ ಇಂಪಾರ್ಟೆಂಟ್ grooves. One, it is the major grooves. This is what we will call it as a major grooves, and this is minor grooves. So if one major groove to the another major groove, it will completes it one 360-degree angle. One complete 360-degree angle alli turn adaga DNA. Okay, so what is the measurement? What we will call it as, that is a 34 angstroms. That is a distance. The distance, one major groove to the another major groove. Okay, so this is one minor groove to the another minor grooves. Okay, one minor groove in the another minor groove. So that is what? That is a complete one turn, we can say. So what we will call it as the 34 angstroms ಅಂತ ಹೇಳ್ಬಿಟ್ಟು ಹೇಳ್ತಿದ್ದೀವಿ. And between these two 34, these two one complete turn, so we will see that is a 10 base pairs. 1, 2, 3, 4, 5, and here also 1, 2, 3, 4, 5. So in one complete turn, we will see 10 base pairs. Okay, in one complete turn, we will see that how many nucleotide base pairs are there. That is a 10 base pairs. Okay, 10 base pairs ಗಳನ್ನ, nitrogenous base pairs ಗಳನ್ನ ನಾವು ನೋಡ್ತೀವಿ. That is a adenine, thymine, guanine, cytosine. Like this, that is a base pairs if it is attached. Okay, so very next, that is the distance between the two consecutive base pairs. The distance between the two consecutive base pairs, this is the 34 angstroms. So two consecutive base pairs in the sense, this is one base pair, that is the A and T here, and G and C here. These two, it will the distance, how it will measure. So what is the measurement here? That is the distance between two base pairs is that is a two consecutive base pairs. This is one base pair, this is two base pairs. That is a 3.4 angstroms. So complete one turn, it is a 34 angstroms. Distance between the two base pairs, it is the 3.4 angstroms. Okay. The last 2 points I will finish up. That is the last 2 points are, that is the DNA helix. There are different types of DNA are there. A DNA, B DNA, C DNA, D DNA and the Z DNA. So based on the twisted helical model, okay, so they are whether it is runs clockwise or anti-clockwise, okay. So like that, based on they are classified A DNA, B DNA, C DNA, D DNA. But what we are going to understand in this syllabus, what it is prescribed, it is a B type of DNA. That is which type of DNA? B type of DNA. So this B type of DNA, it is runs clockwise. Clockwise in the sense clockally ನೀವು ನೋಡಿದಿರಾ. So like this it will be runs. ನನ್ನ ಕಡೆಯಿಂದ ತಿಳ್ಕೊಂಡು ನೋಡಿದಾಗ so ಈ ರೀತಿಯಾಗಿ ರನ್ ಆಗುತ್ತೆ. So this DNA it is runs like this. That is a clockwise DNA. That is a clockwise B DNA. B type of DNA at last. Okay. What is the skeleton of what? This DNA. The skeleton of the DNA is nothing but what? Which will gives the support, which will gives the structure to the DNA. Okay. So ಹೇಗೆ ನಮ್ಮ ದೇಹಕ್ಕೆ ಸ್ಕೆಲಿಟನ್ ಅಂದರೆ ಮೂಳೆಗಳು ಒಂದು ಸ್ಟ್ರಕ್ಚರನ್ನು ಕೊಟ್ಟಿದೆಯೋ ಅದೇ ರೀತಿಯಾಗಿ DNA ಒಂದು ಆಕಾರ ಬಂದಿದೆ ಅಂದರೆ ಒಂದು ಸಪೋರ್ಟಿವ್ ಆಗಿದೆ ಅಂದರೆ so the skeleton of the DNA it is nothing but what? That is a phosphate group. Okay, the skeleton of the DNA it is that skeleton is phosphate group. Phosphate group is the skeleton of the DNA. Okay. So this concludes what? That is a detailed discussion regarding about the structure of the DNA. I hope you are all understand. I repeat once again. That is the DNA it is a two-stranded, that is a double-stranded structure. Each strand it is made up of, that is a nucleotides. Okay, the polymer of nucleotides. So they will runs anti-parallelly. So based on the ends in the opposite direction. Okay. So they runs oppositely and that's why they are anti-parallel, and the purines always pairs with the pyrimidines. That is A bonds with a T, that is adenine pairs with the T with the formation of the 2 hydrogen bond. Guanine pairs with the C with the formation of the 3 hydrogen bonds. And the diameter of the two strands between the one strand and the another opposite strand, there will be a distance, that what we will call it as the diameter. It will measures about the 20 angstroms, and one turn it will measure what? That is a distance between one major group to another major group. Okay, 34 angstroms, and the distance between the two consecutive base pairs, it is the 10 base pairs. That is a one nitrogenous base pair to another nitrogenous base pairs distance. And also the DNA it is a clockwise, the B type of DNA, and it will runs clockwise, and at last the skeleton of the DNA it is the phosphate group, which will forms as the phosphodiester bond in the nucleotide of the heteropolymer of the DNA. I hope you are all understand, dear students. So this concludes what? That is the structure of DNA helical model of the DNA. So how these DNA is studied? By the observation of the X-ray diffraction technique. Okay, so X-ray diffraction, they will take this photograph and they have studied these all these points. Okay, so in the next video we will discuss the next concept. Thank you.